This is part 2 of our look at sound, the auditory system, and pitch. Since we've already taken a look at sound, I want to move on to the auditory system, and specifically the structure of the ear. And we're going to start with the outer ear. Now this is a complete diagram, but we want to focus on the outer ear. And when I say that, you're probably going to think of that fleshy, cartilaginous bit of tissue hanging off the side of your head, which is technically called the pinna. Now, since I have this diagram up, I am going to go a bit more deeply into this. The next component is the auditory canal, or the external auditory meatus. Okay, it is a tube, and it is a tube that's closed on one end. That's going to be important coming up. Next, this takes us into the middle ear past the tympanic membrane. We have the ossicles, which are 3 small bones. They're actually the smallest bones in the human body, and they are the same size today as they were on your birthday. On your birth day. Yes, that's right, they do not get any larger. Finally, we go to the inner ear, where there's a complex looking structure here with a bunch of different components, and I'm going to leave that till a little bit later. All right, let me pull some things out of this. So of course I did start with the pinna, that is the ear on the side of your head, and you'll notice that it's not smooth, it's kind of lumpy and bumpy. Those lumps and bumps and grooves actually serve to channel some sound waves of certain frequencies into your ear canal, or the external auditory meatus, which is about 3 centimeters long. It does serve a protective function in a number of ways. You can't really stick your finger in your ear and poke your eardrum. Don't try and prove me wrong. Also, don't go sticking Q-tips into your ears. You know what it says on the package of Q-tips? Don't stick it in your ear. And you know what happens to thousands of people every year? They stick it in their ear, and then they have to go to the emergency room because they've punctured their eardrum. Don't go shoving things in there. So it does protect some of the delicate, sensitive structures because it's kind of deep in your head. There's also another protective function. The ear canal does produce earwax, which I know is kind of gross. And what it does is it works as an insecticide. And you thought the earwax was gross. So even if a bug does happen to get into your ear canal, it'll be trapped in that kind of sticky wax and die in there. Okay. Kind of makes earwax seem not so gross after all, at least not in comparison. Now, I did mention the ear canal is a tube that is closed at one end. That is really important because it's kind of like having a water bottle. So what happens is sound waves go into your ear canal, they hit your eardrum, and then some of them bounce back out again. Now, if they have just the right wavelength, the ones coming out are going to be reinforced by those same ones coming in, and that's going to increase their amplitude. Now, this happens mostly to frequencies between about 2,000 to 5,000 hertz, and this property is called resonance. You can experience this kind of yourself if you get a bottle and you blow across the top of it. Now, you have to make a little bit of noise, like white noise, with your mouth, with your lips, but then it should work. So this is, I have a bottle. This is definitely not a wine bottle. Okay, I got some pretty good resonance there. Here's definitely not a beer bottle. This could be a water bottle for all you know. Let's try this. Okay, so that's a very different tone that you hear. So those 2 bottles, because they have different sizes, have different resonant frequencies. Find some bottles around your home and try it yourself. Okay, I did mention the eardrum, or the tympanic membrane. That's its technical name. It is basically a bit of skin that is stretched tight to seal off the inner ear from the outer ear. Now, when that is struck by sound waves, it vibrates, and it passes along that vibration to the next series of structures in line. So it transmits sound to the middle ear. I'm going to keep this image up and continue talking about the middle ear. So the next structures we have right past, actually connected to the tympanic membrane, are, well, three structures. The ossicles, I mentioned those. They are the three smallest bones in your body. There are three bones. They do move with respect to each other. They do have names, and yes, you need to know the names and know them in order. They are the hammer, the anvil, and the stirrup, or the malleus, the incus, and the stapes. Now, they are held in place by some muscles in the middle ear. And the order is pretty important. On the one end, the malleus is connected to the tympanic membrane, and at the other end, we have the stapes connected to the oval window on another structure that I will get to. Now, we are actually exquisitely sensitive to sounds, even though you might not believe your sense of hearing is that acute. At 0 decibels, the bones of the middle ear move less than the diameter of a hydrogen atom. And you can still pick up that sound about 50% of the time. That is pretty darn sensitive. Let's just focus on the ossicles a little bit. Now, one of the things that the ossicles do is they take the vibration that's on the eardrum, which is relatively large, about 0.6 square centimeters, and they concentrate that vibration on the oval window at the other end, which is way smaller. So what that does is it actually increases the pressure by about 20-to-1, which is a lot. The second thing is the ossicles do move with respect to each other, and they move with a lever action. And this further increases the vibration by 1.3 times. So it's pretty clear that the function of the ossicles is to increase pressure. But why would they have to do that? The answer is related to what you might have experienced if you've ever done a belly flop into a swimming pool. Has anyone ever convinced you to do a belly flop? Hey, it'll be fun, you should try. No, it's definitely not fun. You start your dive in the air and things are going fine until you hit the surface of the water. And then it's quite unpleasant. So far, this is kind of similar to what's happening with the ear. Both the outer and the middle ear are filled with air. The inner ear, however, is filled with a fluid called cochlear fluid. Now, when you hit the surface of the water, that's pretty unpleasant because the water is going to be actually reflecting a lot of your kinetic energy back at you, which is really not a heck of a lot of fun. Now, the same kind of thing happens to the sound waves as they hit the solid structures and the cochlear fluid. A lot of that vibration is going to be reflected back out of the ear and basically lost. So to compensate for this upcoming loss of pressure, the ossicles work to ramp up the pressure at the last possible moment. So good job, ossicles. There's one more structure in the middle ear I want to talk about. Going back to the diagram, it's not explicitly labeled, but it's the eustachian tube. Now this actually helps to equalize pressure with the middle ear and the outside world. So if you're ever on an airplane, for example, and your hearing kind of goes a little bit fuzzy and you can't hear, so you might yawn or swallow or chew gum, for example, and your ears will kind of pop and go back to normal. Sometimes this happens if you take a really fast elevator. So what happens is the pressure in your middle ear is different from the pressure outside, and it kind of fuzzes up your hearing until you open up your eustachian tube, and that equalizes the pressure again. All right, moving along, we now come to the inner ear. So it's actually taking us quite a while to get to the actual receptor structures, and they are somewhere in the inner ear. Before I get to that, though, a slight detour through the semicircular canals, which I will be coming back to in a later lecture. The semicircular canals don't have anything to do with audition or hearing per se. They are involved in balance. It's part of our vestibular sense. This is not the greatest diagram. You can't see all three of them. There are three of them, and interestingly, they are arranged at right angles to each other. So these can help you sense movement in any direction. Now, the semicircular canals are kind of part of this other pink structure here. It's called the cochlea. It is kind of snail-shaped, and it's been kind of cut away here. It is filled with cochlear fluid, as I mentioned. It has 2 3/4 turns, and if you do slice through it, you see it is divided into canals. You can actually see there are 1, 2, 3 canals there. Yes, the canals are going to be important. We have the scala vestibuli, or the vestibular canal. Sorry for the Latin, but there is a lot of Latin that is applied in audition. There is also the scala tympani, or the tympanic canal. Now, these do get their own separate names, but they actually turn out to be kind of the same, because one of them is kind of going in, and then they kind of meet up, and the other one is kind of coming back out again. They're connected at the apex end of the cochlea at what's called the helicotrema. Now, these, are important. They do need to be there, but really the most important canal for our purposes is the scala media, or cochlear partition. Now, this particular canal is created by a couple of membranes, the basilar membrane and Reissner's membrane. Okay, I promise we are zooming in eventually to the receptors for audition. The scala media is really important because it contains the organ of Corti, which, let's just go to some pictures now, take a break from all this Latin. Okay, so here's a slice through the cochlea, and we can see the scala vestibuli, the scala tympani, and the scala media. If you kind of zoom in on there a little bit, you can see, well, there's a bunch of junk, okay? And that is part of the organ of Corti. Kind of hard to see, so I'm going to zoom in on that a little bit more. I'm going to break that out. There we go. Okay, so we've got a couple of membranes here. One membrane is basically all of this kind of tan colored structure, and then the one kind of lying on top of it, kind of semi-transparent and kind of purplish here, is the tectorial membrane. Now, the basilar membrane is really important. I'm going to be talking about it a lot in the rest of this lecture, okay, because the basilar membrane contains, finally, some of the receptor cells. We have the inner hair cells here, and then we have outer hair cells. Yeah, we've got two different kind of areas here and two different kinds of cells, interestingly. Okay, so to break down the organ of Corti, again, it's that kind of purplish tectorial membrane which overhangs the basilar membrane, and it's the basilar membrane that contains about 15,000 hair cells per cochlea per ear. Maybe there's a parallel between audition and vision. Wouldn't that be neat? Because in vision, remember, there are two kinds of receptors in the retina. It's not going to be exactly like that, unfortunately. So let me break down the different kinds of hair cells. Okay, so there are the inner and the outer hair cells. They differ in terms of their number. There are many more outer than inner hair cells, again, per cochlea. As I showed you in the diagram, the inner hair cells, and this is the cells themselves, the inner ones are aligned in a single row, the outer ones 3 to 5 rows. And in the picture I showed you, they had 5 rows. Now, it was really hard to see in that diagram that I showed you, but the hair cells are called hair cells because they have little, hairs sticking up out of them. Okay, if you want to be technical, and you probably should be, they're called cilia. The inner hair cells have about 40 to 60 of these cilia per cell. For the outer hair cells, again, big difference, way more, 100 to 120 per cell. And that's not it. The cilia in the inner hair cells, so the little hairs, are arranged in straight lines, but for the outer hair cells, they're shaped like V's or W's, which is kind of strange. And last, but totally definitely not least, the inner hair cells are sensory. That is, they are picking up the sound waves, the vibrations. The outer hair cells are not. I'm going to be kind of vague here. I'm going to call them supporting, and we're going to get to exactly what they do coming up. But first, some scanning electron micrographs. Okay, so we have the IHC, that is the inner hair cells, and yeah, the cilia are kind of in straight lines. Okay, just pretend they're straight lines, okay? And here are the outer hair cells, pretty clearly kind of V's or W's, very different shape from the inner hair cell cilia. and there's some colored ones, and that's really zooming in a heck of a lot, and you can see those cilia sticking up. Okay, so these are some of the frontline structures, but they connect to further cells down the line, neurons. So we come to the spiral ganglion cells, and we have 30,000 of these, again, per cochlea. And interestingly, there are a couple of different types. 95% are called type 1. Now, pay careful attention here because it's really easy to get mixed up. For the type 1, there are between 3 and 15 of these type 1 spiral ganglion cells that go to each inner hair cell. These spiral ganglion cells are relatively large and myelinated. The remainder, about 5%, are type 2, and take a look at this pattern of connectivity. One type 2 spiral ganglion cell goes to 10 outer hair cells, so kind of the opposite pattern of connectivity. These spiral ganglion cells are relatively small and relatively slow. The transduction process is how we encode the information that's contained in, say, the energy or the chemicals. So when it comes to audition, just to kind of summarize where we've gotten to so far, we've started off with some vibration in the environment, and that causes a pattern of vibration on the eardrum. This causes it to be transmitted to the ossicles, then to the oval window, and then to the vestibular canal, the cochlear partition, which contains the cochlear fluid, and this finally presents the vibration to the basilar membrane. Yeah, it takes quite a bit to finally get to the hardware that's actually carrying out the perception. And just a note about the membranes. The basilar and the tectorial membranes, they don't move up and down. They move laterally, that is, side to side, with respect to each other. They've produced a shearing force. Now, when that shearing force is produced, what it does is it bends the cilia, specifically of the outer hair cells. Now, the outer hair cells turn out to be embedded in that kind of overhanging, that transparent purplish tectorial membrane. Now remember, the outer hair cells are not involved in any sensory function whatsoever. So what do? I'll get to it. Patience. Let's turn to the inner hair cells. Now it turns out the cilia of the inner hair cells, they too are bent when there's a vibration, but they're bent by the flow of the fluid that surrounds them. Here things get a little bit more complex. I'm going to have to zoom in on this a little bit. The tips of the cilia are linked by a stretchy fiber called actin. When there is a sound, the inner hair cells will bend and move, and the cilia are going to pull on the actin. The actin is going to stretch as much as it can until it reaches its limit, and then when it can't stretch anymore, it kind of pops open a trapdoor in the tip of the cilia. And when that happens, again, imagine this is all surrounded by fluid, the trapdoor opens and potassium ions are going to flood into that inner hair cell. This is going to start the neural signal by starting to release glutamate. That's the first thing. I know it's kind of hard to imagine, so here's a picture of that. All right, so in this case, on the first example over here on your left, they're not really moving too much, and the actin hasn't stretched too much. Trapdoors are not open. But in the second example, there's a strong flow of fluid. It moves those cilia over. The actin stretches as much as it can, and it can't stretch anymore, and it pops open that trapdoor, and that allows potassium to enter into the hair cells. I have an animation as well. These animations are limited to what I can kind of find on the web. So this one, for some reason, it uses fish. I'm going to reset it so you can get the full effect of this impressive animation. Okay, so we start with our fish, and we're going to zoom in. Okay, it looks a little different than the diagram I showed you. Hey, it's a fish. Okay, so this is the resting state. There are some of these channels open. We can see the trap doors and the actin fibers as well. Let me create a vibration. Okay, so there's a second fish swimming by. And now we get depolarization. Those trapdoors are fully open. And they're going to swing the other way. And they're going to be completely closed. We get hyperpolarization. And then back to the resting state again. Impressive animation. Okay, it's whatever I could find. So what we need to do now is continue to trace the path that the information takes. So we've had some environmental events, some pressure waves. It has turned into sound. It has been detected by an auditory system. It's gone into the ears, literally. It's gone through the ossicles. It's gone onto the basilar membrane, and it's activated those inner hair cells. Now what? Okay, we're going to continue on. Pardon my drawings. Some of these are my drawings. So these spirals, these are the cochleas. Now, I'm not going to show you absolutely everything from both ears. I just want to focus a bit on one ear. This diagram is kind of twice as complex as it needs to be because it shows the structures bilaterally, so on each side of the brain. So it is pretty schematic. I didn't want to draw it and make it look really nice, obviously. Okay, and I do not expect you to completely memorize this diagram. There is a lot going on. So we start with a stimulus. We get a signal coming out of the cochlea, and this arrow right here, let me just say, that's comprised of the axons of the spiral ganglion. So this arrow here, and this one over on the left side as well, these are the auditory nerve. Okay, so they go to the cochlear nuclei, these large structures, again, bilaterally, and it's a direct connection. But from there, things start to turn into a plate of spaghetti because the cochlear nuclei send connections both ipsilaterally on the same side of the brain and also contralaterally to the superior olives in the brainstem. There's also another contralateral connection to the inferior colliculus. It's part of the midbrain. From there, I don't know where to start, but from the superior olives, connections continue to feed forward to the inferior colliculi, and then from there, some information gets shared between the two hemispheres back and forth, and then to the medial geniculate nuclei of the thalamus. And then from there, from these blue regions, they project to the auditory cortex and the temporal lobe. Whew, there is a lot of stuff going on. And you know what? I even left out some connections. There is a really important connection that I left out. I do want to talk about it. And that is feedback connections from the superior olives. They send feedback back to the outer hair cells. What? That's really? Now, this is not like anything we've seen in any of the other senses. There's no counterpart to vision here, not exactly. That there's feedback going to the outer hair cells. Well, remember, I didn't really explain what the outer hair cells were doing. I just said they were supportive. Didn't say exactly what they're doing. I will be getting to that eventually. It's not that I'm trying to keep you in suspense, but that's actually fitting in really well to one of the theories of pitch perception. All right, we can zoom out to the auditory cortex and try to get a sense of the big picture, how the auditory cortex organizes this incoming information. The organization is called tonotopic. That is, if we pick a couple of neurons in the auditory cortex, stick a microelectrode into them, and then present a whole bunch of different frequencies, we will find that adjacent neurons will respond to very similar frequencies. What that means is that frequency is very important to the brain when it's processing auditory information. It wants to preserve that. Not only are there individual neurons that are activated by different frequencies, but they're also physically close together. Like keys on a piano playing similar frequencies are also close to each other. This is pretty important to the auditory system. We have to ask the question, why? Why is that important? There's an image of the tonotopic organization. Now, this information is preserved from the basilar membrane all the way to the auditory cortex. Now, to survey the parts of the brain about 40% of neurons do not respond to an individual frequency. They will be activated by things that don't have a particular frequency, to noise, clicks, and bangs, which means that about 60% of neurons do respond to a certain frequency. Is that a high number, or is that a small number? You can think of it maybe as a pretty large number. 60% of neurons will respond to one single frequency. But under what conditions are we going to hear a certain frequency all by itself out in the wild? The answer is almost never. So why 60%? Well, now we're helped once again by our old friend, you know it, you hate it, Fourier analysis. Fourier analysis is a process that we can do. You can do it with pencil and paper or a calculator. You can take a complex tone and break it down into its component sine waves. But maybe our auditory system is doing exactly the same thing. If we are hearing a complex waveform, maybe our auditory system is doing Fourier analysis, breaking it apart into its component sine waves. And if it weren't doing that, I wouldn't have brought this all up. So yes, it looks like our auditory system actually is doing this Fourier analysis. Now this might seem to be kind of overkill. Like that's a lot of processing that must have to take place. And what's the point of that? Well, again, we have to come back to the different approaches to sensory coding. One approach is a specificity theory, which says that every slightly different thing that you can hear is activating a different neuron. Or it might be a more distributed representation in which a number of things come together. It's the pattern of the responses of neurons that encode a particular stimulus. And it's this latter approach which is probably more efficient. So for this reason, if it is this kind of pattern approach, we won't need to have a specific neuron set aside that will respond when someone plays middle C on a violin, and then another neuron set aside for middle C on a piano and middle C on every single instrument, and then every other frequency as well, frequencies that don't even correspond to notes. Just like we would have to have a separate neuron for every different tree that you could possibly see, we'd have to have a separate neuron set aside for every slightly different sound that you could hear. That's really not very efficient. Again, it is 60%. It's maybe only 60%. We might have some neurons set aside to detect specific sounds. In human beings, not very many have been found so far, but for other animals, like squirrel monkeys, for example, we know that they have squirrel monkey sound detectors. And that kind of makes sense. It would be adaptive for them if they had that set aside. It is particularly relevant to squirrel monkeys. We don't have squirrel monkey sound detectors. That's not particularly relevant to us. I'll stop part 2 here.